Good evening, everyone. My name's Josh. I manage the outreach activities for the Australian Research Council Centre of Excellence in Exciton Science. And I'd like to start by acknowledging the traditional owners of the land from which I'm connecting to you from, the Wurundjeri people of the Kulin Nations, and pay my respects to their elders, both past and present. And as we're here to celebrate science and technology, it's only fitting that I also acknowledge Aboriginal and Torres Strait Islander peoples as the first scientists. But welcome to our second and final public lecture for National Science Week. I hope everyone's made it to at least a couple of other events, but if not, welcome to your first National Science Week event. Tonight, we'll be hearing from Dr. Nicholas Kirkwood from Exciton Science and the University of Melbourne, as well as Dr. Valerie Mitchell from the Australian Nuclear Science and Technology Organisation, otherwise known as ANSTO. But before we begin, we're going to do a little live poll as a bit of an introduction to what we're going to be talking about today. So if everyone could jump onto that Slido link, put in that code if you need to, and I'll give you about 20 seconds to do that if you haven't already, just in case you need it. That's it there as well. And I'm gonna just share that part as well. So hopefully this works. All right. So it looks like we've got about 24 people here or who have voted. So not all of you, but that's all right. And I asked the question, which of these would you rather have? Solar panels that are better at converting light, seems to be in the lead at the moment. Lighter, flexible solar panels coming second. Reliable ways to detect dangerous chemicals, still very important. And more energy efficient lighting is all the way at the end there. But, you know, still with some votes. Now, the reason I ask this question is because here at Exciton Science, we're actually working on all of these things. We're spread out across 5 universities. And the centre consists of chemists, physicists, engineers, all working on different materials and how they interact with light. So these are actually, all things that we're kind of helping develop and bringing to life. All right, so let's move on to the second question. Hope that was very exciting for everybody. All right, will that jump over for me? Great. Hopefully an easy enough question to answer. Have you ever had a COVID-19 test? I think for the most part, this should be one of those swab tests as well. 30 people. This is actually quite interesting to me, given there's actually a considerable chunk of people who haven't had to yet get a COVID test. This is maybe an overshare, but I personally have had, I think 4 now, but I'm a bit of a hypochondriac. All right. In total, Australia has actually done 28,776,897 COVID-19 tests, which is quite a lot. And I have a feeling there's going to be plenty more, unfortunately. And while some of us did that pivot to online content, like this public lecture, some of our researchers actually pivoted to helping create integral components for these COVID-19 tests. And that's something that we'll be hearing a lot more about tonight. All right, on to the next question. And the final one. If it wants to jump over. Here we go. What's the largest amount of people you've had to cook for? 1, between 2 and 10, between 11 and 50, between 50 and 500, or between 500 and 1,000. One is obviously the loneliest number, so I hope no one has had to only ever cook for one person. And of course, as soon as I say that, we get someone answering that way. All righty, cool. And I take note that no one has had to cook for between 500 and 1,000 people. And you may be wondering why I've asked this question. Well, that will become apparent in the not too distant future, hopefully. Hopefully Nick will answer this question and why it's important. But without further ado, I'm going to stop asking silly questions. I'm going to hand over to our first speaker for this public lecture, Dr. Nicholas Kirkwood.
Thanks, Josh. Thanks for the introduction. Just going to share my screen. Okay, great. Okay, thank you, Josh, for the introduction and the little quiz at the start there. I will attempt to make it clear why we're asking how many people everyone's had to cook for, I promise. So welcome everyone to this half of our talk. I'm going to be talking to you today about how some of the solar energy technology that we're developing at Exciton Science is actually helping to detect COVID. So, cool. So in order to tell this story, we have to first rewind back to January 2020 when the pandemic had not yet arrived in Australia. So my research was then and still is focused on making and understanding nanoparticles. So what is a nanoparticle? The most important thing is that a nanoparticle is really, really tiny. So a nanometer is a billionth of a meter. And what this means is that if we take thousands and thousands and thousands of nanoparticles, they would only fit just across the width of a human hair. So one of the interesting things about this is that you can't even see them, even with the world's most powerful light microscope. You cannot actually see a nanoparticle, they're just too small. So in our lab, we actually grow nanoparticles inside flasks. So in this photo in the middle here, you can see a glowing liquid, and that liquid is actually full of billions and billions of little nanoparticles. And although we can't see them, we can see that they're glowing, and that's because of the properties of this particular batch that we've made. And we can make nanoparticles out of many different materials, such as fluorescent materials. So this is actually a series of different nanoparticles in vials, and all of the nanoparticles in each of these vials are the same material, just different sizes and shapes. We can make them out of polymers, and there's kind of a rainbow effect going on here. These nanoparticles are made out of gold, and this kind of illustrates the point that nanoparticles actually can behave really differently and uniquely compared to their larger particles made up of the same material. So none of those things actually look like gold, but each of those vials is containing gold nanoparticles. And so at Exciton Science, we're really interested in using the unique properties of nanoparticles to convert light into electricity. And that's basically solar energy. So we're developing, for example, next generation solar cells, things that we're calling solar concentrators or luminescent solar concentrators. This is a picture of one here in the middle of the screen. And you can see that there's a red bit of plastic, and that bit of plastic, or it could be glass, is filled with billions and billions of little nanoparticles that are able to absorb the sun's energy and transmit it to the edges of that sheet. And that's why the edges are glowing so strongly. And it turns out we can take this kind of technology and use it to, for example, make something called a solar window. So we're working with the company Clearview to develop solar windows, which could be, for example, used to make a greenhouse which can power itself, or even maybe a skyscraper which can power itself. As we all know, though, around about March 2020, there was a sudden and dramatic change in that the COVID pandemic arrived in Australia. And for research science, this was a real jolt to a stop, because we were actually at Melbourne Uni, for example, locked out of our labs. We weren't allowed to go in during the lockdown. This was a bit of a change of pace for us, suddenly having to work from home and wondering how we could what we would do as research scientists during the pandemic. And at the same time, however, there was a sudden and dramatic need for COVID tests. So when COVID hit, obviously there were no COVID tests being conducted in Australia, but over time, the number of tests that we're needing to do is now in the hundreds of thousands per day. We do have some local test kit manufacturers in Australia that assemble COVID test kits and sell them. But it turns out a lot of the components of these test kits are still sourced from overseas. And the one that I'll be talking about today that is particularly interesting for us is called a magnetic silica nanoparticle. And don't worry, I'm going to explain in more detail what that actually means later on. But the government was really worried that because a lot of the components for these test kits are made overseas, that if there was supply chain disruptions or price increases, there would be an inability in Australia to do testing. So they really wanted to make sure that everything could be made in country if necessary. And so the government put a test kit task force together, and they actually contacted our research centre, Exciton Science, to see if we could create or help create a local supply of these nanoparticles. And the original target was enough for about 100,000 tests per week. And because my boss is a kind of we can do it person, he said yes, which means we were very quickly meeting on Zoom and scrambling to figure out how we could achieve this. But before I continue with the story, I guess the questions that you might have and that we need to answer here is, what is a magnetic nanoparticle, and why would we even need to use one in a COVID test anyway? So what is a COVID test and how do they work? So this first type of COVID test I'll talk about is a PCR test, or polymerase chain reaction. More commonly, we call these swab tests, and these are the tests that, I don't know, what was the number, about 80% of us, according to that poll Josh just ran, will have had if you have been tested for COVID in Australia. And the reason this is the main test that we use in Australia is because it is really, really accurate. There's some downsides. It's slow. As we all know, it can take hours or usually sometimes days to get a result back. And they're reasonably expensive to do as well. But this is the gold standard for COVID tests. On the other side, we have what we call an antigen test, or more commonly a 15-minute or a rapid COVID test. And these kind of look and actually are the same technology as a pregnancy test. And these are not officially used by the government in Australia yet, although they are discussing it. They are less accurate, and that's probably the main reason why they're not the primary means we use to test for COVID, but they're getting better. These, however, are really, really fast. So the tests are in minutes. The result arrives in minutes, sorry. And the result is done, at the site of the test. And it's also really cheap to do a test with these things. So there probably will be a lot more scope to use these as the pandemic evolves. Actually, really interestingly, I'm actually going to mostly talk about the PCR test today. That's where these magnetic silica nanoparticles are used, and that's what this story is mostly about. But just briefly, there's actually a really strong link between this antigen test and solar energy technology, as it turns out. So it turns out that the same fluorescent nanomaterials that we're using for those solar windows that I mentioned earlier can also be used, and are also being used, by a spin-off company from Exciton Science to make a really innovative new COVID rapid or antigen test. So I'll leave the details of that story for another time when these tests are more developed. But I think this is a really good illustration of how powerful nanotechnology can be, that one material can be used for multiple different applications that seem, on paper at least, completely disparate. So back to that PCR test, which I said would be the main focus of the talk. In order to understand what happens here and why we need to use nanotechnology, first I'm going to ask what's in a swab. So if you're unlucky, the back of your throat, if you have COVID, will have COVID viruses there. And that's what they're looking for when they take a swab. Now, COVID is just a tiny little package, which is full of RNA, or ribonucleic acid. So RNA is just the genetic information of COVID. It's the same thing as DNA in a human or animal body, just that RNA is a single strand of genetic information, whereas DNA is a double strand of genetic information. So when we take this swab, not very pleasant, but this guy's just had his swab collected, the test person will then put the swab in a solution which actually breaks apart all of the all of the cellular material that was collected in that swab. And what this means is we get a kind of like a swab soup, let's call it, which has COVID RNA in it, if you were unlucky enough to have COVID. We'll also have lots and lots of DNA from the patient's cells, all of the cells in the back of their throat and mouth, maybe also some bacteria and viral DNA from other bacteria and viruses you might have had. There's also going to be other stuff in there, because remember, this swab is collected from the back of the nose and the throat. There will be all sorts of stuff there, fats, proteins, bits of broken up cells. It's from your throat, maybe even the bits of dinner that you had last night, but maybe we won't go into the details there. So the PCR test itself is just looking for that COVID RNA. If there's any COVID RNA, the PCR test will be able to pick it up and it will just give a positive test result. The problem is that this test actually doesn't work if we've got too much of that other stuff present. The reason is it kind of gunks up the machine. So in order to actually run a PCR test, we have to somehow separate RNA and DNA in that solution from all of the other stuff. And that's a really tricky problem. These things are all far too small to see with the naked eye or to manipulate with any kind of tweezers or anything or filter. So this is actually a really big problem. And actually the solution is to use magnetic silica nanoparticles. So not a particularly imaginative name for this type of nanoparticle, because they have a magnetic core that's typically made of iron oxide. That's the same material that you might see in a fridge magnet. And it's coated with a silica shell, silica is actually the same as glass. Now remember, these are nanoparticles, so we could fit thousands and thousands and thousands of this in the width of a human hair. So they're really, really tiny. You can't see them with your naked eye. But what we can do is dissolve them in water. And this vial on the right here is actually full of magnetic nanoparticles, and that silver thing is a magnet. And you can see when we put the magnet on the side of the vial, all of the nanoparticles that were dissolved in the water are separated to one side, and the water is left alone. And so now we have a way, because these are magnetic, we can move them around with a magnet. So we have a way of separating them from the rest of the liquid. So then why the silica shell? So if we put these magnetic silica nanoparticles into our swab soup, remember we wanted to separate that DNA and RNA from all the other stuff. It turns out that under the right conditions, DNA and RNA will stick to silica, which means they will stick to our magnetic silica particles because they're coated in silica. And now, because we have a magnet inside them, we can move them around like I just showed in that video. And you might see where this is going. We can use that magnet to pull the particles and the DNA and RNA attached to them away from everything else. So if you take a look back at that video that I showed you, I'll just wait for it to loop. So we get back to the starting point, and now we put our magnet there. And now what we're seeing is that the other stuff will be left in the liquid, and our DNA and RNA is collected into that magnetic silica pellet where the magnet was. And then we can simply chuck away all the other stuff that we didn't want. Now we take a new liquid, and this is a special liquid which is going to unbind RNA and DNA. So unstick them essentially. We put the particles back into this liquid, and now the DNA and RNA will float free. Now we can remove those nanomagnets using a magnet, and we have what we wanted. We have a clean solution which only has DNA and RNA in it. And this is what we put in the PCR machine. And now if this person was unfortunate enough to have COVID, it will test positive for COVID. Now I should say there's other ways you can purify DNA and RNA, but this is by far the quickest and the cheapest way to do it. And remember, we're doing hundreds of thousands of tests a day in Australia, so we really need a cheap and a quick way to test for COVID. So that's kind of the science or the theory behind why we need magnetic silica nanoparticles in a COVID test kit. How do we go about making a local product? So companies, overseas companies, are already making these types of materials, but they don't divulge the methods. The private companies are not going to tell us how they're making it. But we really needed to be producing it. So what we realized was that the experience and resources that we had gained as researchers making nanoparticles for solar energy applications, we could use all those same skills and chemistries to produce magnetic silica nanoparticles in Australia. And over the last year and a half, myself and my team have made well over 500 experiments to test and optimize magnetic silica nanoparticles to create a viable product that will work in COVID test kits and can be made in Australia. We haven't done it all alone. We've had lots of help along the way. So, for example, at Monash University, the electron microscopy center there were able to take absolutely amazing images of our nanoparticles. And this is really critical because if you just, let me pull up a laser pointer here. If you think of nanoparticles, I mentioned they're too small to see with even a light microscope. So we actually have to use these very special electron microscopes to image them. And this is the sort of stuff we get. And you can see here, these are our magnetic silica nanoparticles. The dark spots in the middle are the magnetic cores, and then the gray stuff on the outside is the silica shell. And so we can compare different batches and see how what we've made, how it looks, and use that to improve what we're doing. Of course, we've also had to work with a test kit company who make test kits, Genetic Signatures in Sydney, and they've been checking all of the batches that we make to actually verify that they really work in COVID tests. There's obviously no point making them if they don't work. And so just to show you kind of some of the journey we've been on, this is an early prototype. This is clearly magnetic. These are magnetic nanoparticles dissolved in water. The problem with this is that the magnetic nanoparticles follow the magnet, but so does the liquid. So remember, we have to be able to separate the magnetic nanoparticles from the liquid. And that's really cool what they're doing there, but it's actually not what we want. So that approach didn't work. In the middle now, we have another batch, another prototype, and this one is too slow. So they're magnetic. There's a really, really big magnet now on the side of the vial there. And you can maybe just start to see now that those particles are moving across towards the magnet. The problem is that it's too slow. And actually the main issue here is that there's a risk that some of the magnetic nanoparticles don't make it. And then we might get a false negative because maybe there was some COVID left in the solution that we get rid of. Okay, this is a much more recent product that we've made, and you can see here fast, clean separation between the magnetic nanoparticles and the water. And they're also really easily resuspended afterwards, which is also very nice. So that's kind of the journey we've been on to create a product that works in our lab. And then we faced the biggest problem of this project by far, which was one of scale. And now this is where the part of the cooking question comes in. If I ask you to make pasta sauce at home for 2 people, this is the one I made the other night. It's not a big problem. Most kitchens are capable of doing this. Most cooks are capable of doing this. But how about if I ask you in your home kitchen to make enough pasta for 1,000 people in a single go, in a single batch? I suspect the answer is you can't, unless you have a very different kitchen to mine. And this is the problem we faced. I mean, this photo I've now put up on the right of the screen is a typical reaction that we use to make magnetic silica nanoparticles. This is the equivalent of our kitchen. And the problem is that a typical research lab reaction, we might make, let's say, about 1 gram of magnetic nanoparticles in a single reaction. But if we need to make enough for 100,000 COVID tests a week, that's 500 grams approximately of nanoparticles per week we need to be making. And we just simply cannot run 500 of these reactions a week. We don't have enough time. And so we needed to scale up by a lot. And we just don't have big enough flasks and heating apparatus in our lab to do that, in the same way that you wouldn't in your home kitchen have a big enough pot nor a big enough gas burner to make enough pasta for 1,000 people. So we turned to local industry, and luckily we found a couple of companies who were willing to help us and make it possible for us to see if this reaction could be scaled up to produce a product that works but can also be made at the appropriate scale. And so this company, Scale Organics, made available what's called a pilot reactor to us. This is me standing in front of my very first ever 70-litre reaction, well over 70 times bigger than what I'd probably ever done before at this point in my career. We were actually able to work with these companies to produce batches that were making over 500 grams of nanoparticles. And that's, remember, more than enough for 100,000 tests in a single go, which is pretty incredible and really bizarre to go from making something that fits into a tiny vial to making something that we actually realized we didn't have a bucket big enough to store it all in, and we were having to run down to Bunnings and buy lots of giant buckets in order to store our product. Obviously, our methods became more sophisticated as we went along, but needless to say, early on in the project, I think Bunnings were quite confused why there was a whole bunch of nerdy scientists running into Bunnings every other day in the middle of a pandemic, really, really interested in buying lots of 20-litre buckets. Buckets aside, this was an absolutely massive effort and months and months and months of hard work from a large team of people within our centre and outside of it as well. But we can now make magnetic silica nanoparticles in Australia, and we can make lots of them in a single batch. And critically, we can show that they really work in COVID test kits. So this is now, let's say, a viable product. The next steps, we're working on setting up production, commercialization, and all of the other things that will come with that. I did mention this is a team effort. It really has been a huge effort. So I'd just like to thank all of the people involved. So, for example, all of the students and staff within our research lab at Melbourne University and within Exciton Science in general. The Australian Government Research Council who've funded our research centre, and really I think this project highlights what a well-funded research effort can achieve, and how quickly we can respond to pressing problems. Our industry partners, particularly Scout Organics, Tritech, AMT, and all of the people there who've helped us, particularly with the scaling up aspects of this. Monash University, Genetic Signatures, another industry partner, Dr. Sasha Midden there, who really, really helped doing thousands of COVID tests for us to verify our products would work. And last but definitely not least, you'll notice the synchrotron down here. So why was the synchrotron involved, and what do these researchers have to do with this problem? Well, Dr. Valerie Mitchell is going to talk next, because this wasn't all smooth sailing the whole way through. It turns out some batches actually slowly performed worse over time, and we didn't know why. We were getting some that were working beautifully and others that weren't, and it was really a bit of a head-scratching moment for us. And we were fortunate that the Australian Synchrotron, which is an amazing bit of equipment, if I can call it that, down in Clayton, was able to help us answer this question. So Dr. Valerie Mitchell will speak next about that exact problem and how the synchrotron was able to help us answer it. So without further ado, thank you for listening to my half of the talk, and I will pass over to Valerie now.
Great, thanks, Nick. Just an excellent talk and some really exciting research. So thank you for that. And let's see, I will.
Thanks, Valerie.
Where's the share screen button? I missed it somewhere. Oh gosh. Okay. How's that looking? Good.
Yep, looks great.
Okay, thanks. All right, great. Yes, so my name is Valerie Mitchell. I am a beamline scientist at the Australian Synchrotron, and I work at the X-ray absorption spectroscopy beamline. And I'm going to be talking to you a bit about how the Australian Synchrotron helped out in this project and helped kind of identify some of the factors that were affecting the nanoparticle performance. So, as Nick said, as they were developing these recipes for the nanoparticles, they were getting a little bit of variability. So they were seeing some particles that degraded over time, some particles that were less magnetic. So you can see this nice video over here of two different batches performing differently. And the result of that would be that the particles wouldn't work as well in tests. And so it was really important to figure out what was happening here and why some of these particles weren't working as well. And these are pretty complicated systems. There's a lot of variables. So some of them maybe could have been the size of the nanoparticles, the silica shell. But one important thing to consider would be the composition of these nanoparticles. So they were iron oxide, that was for sure. But it turns out that iron oxide can actually come in several different forms, and depending on the form, and I mean the way that the iron and the oxygen are bound to each other, the properties will differ quite a bit. So here we've got 3 different types of iron oxide. So on the left we have hematite, which is non-magnetic, and in the middle magamite, which degrades slowly over time into hematite, which would be non-magnetic. And on the right is magnetite, which is stable and magnetic. So this is really the ideal composition for these nanoparticles. So there was some question, like possibly there could be mixtures or different types of iron oxide in these nanoparticles that are affecting the performance. But unfortunately, this is really difficult to determine with conventional methods, I guess you would say. So because the iron oxides are all very similar, it's really difficult to pinpoint the differences. But luckily, at the X-ray absorption spectroscopy beamline, that is our specialty, telling the difference between compounds like this and elements that are closely related. So this is our XAS, X-ray absorption spectroscopy team down here, looking successful. All right, so I'm going to start by just introducing kind of the theory behind XAS, or X-ray absorption spectroscopy. So here we have 5 corgis. So dogs can be trained to respond to specific tones. So these corgis have all been trained to respond to different tones. So if we work our way through the musical notes, you'll see each corgi responds to the one appropriately. So this is great. And what's useful about this is that if we then had a mystery corgi, we could likewise just scan through the tones until we got a response. And when we got that response, we'd be able to identify our corgi. So this Corgi identification method works because the corgis respond specifically to tones, but also because we can play one tone at a time. So if we could only play all the tones at once, all the corgis would do their thing at once, and obviously that would be delightful, but not particularly useful for identification. So what does this have to do with spectroscopy? So just the way sound is broken up into different notes, light is broken up into different wavelengths. And just the way all of these corgis respond to specific tones, the elements will actually respond to different wavelengths of light. So each one of these elements will respond very specifically to a certain wavelength of light and none other. So we can identify elements by scanning across wavelengths of light and looking for a response. So here I've kind of got a little cartoon where I'm trying to represent the visible light spectrum with these little waves. But actually the visible light spectrum is not high enough in energy to get a response out of these elements the way we need it to. So we don't use visible light for these experiments. We actually use X-rays. So X-rays are also a type of light, but they're much higher in energy. And they're not visible, obviously. So much like how a very high-pitched tone won't be audible to the human ear, X-rays are outside of the range of human vision. And you're probably familiar with X-rays from the medical fields, from going to the doctor's office and getting the X-ray. The same kind of stuff. The difference is that the X-rays that they use in the doctor's office are pretty weak. They would be way too weak to do the kind of experiment that we need to do to analyze for elemental composition. They also use a bunch of different wavelengths at once. So there's really no way you could do this X-ray absorption spectroscopy with a medical wavelength, or sorry, a medical X-ray. But this is where the Australian synchrotron comes in. So the Australian synchrotron, as Nick introduced, is an extremely powerful source of X-rays. So the Australian synchrotron is located out in Clayton. It's right across the street from Monash University. This is a view of it here. And the synchrotron is a particle accelerator. You may be familiar with the term particle accelerator from things like the Large Hadron Collider is an example, but we don't actually collide anything. So we, colliders will accelerate particles and then crash them into things to look at the results, but we actually just accelerate them to extremely high speeds, because particles moving at extremely high speeds give off light. Excuse me. So when I say high speeds, I mean 100 billion kilometers per hour, which is equivalent to circling the Earth 7 times per second. So these particles are really flying, and they produce extremely strong light. So the light is about 1 million times brighter than the sun. And a lot of the light, most of the light, is X-rays, which we can use for, say, X-ray absorption spectroscopy. So this brings us back to Nick's problem. So in X-ray absorption spectroscopy, what we do is we put a sample in front of the beam. We pass one wavelength at a time through the sample, monitor the signal with the detector, and when we see that signal jump up, then we can identify the material. So Nick wanted to know, what are the commercial nanoparticles made of? What conditions produce the best nanoparticles? And why are some of their samples degrading? Why are some of their samples not performing as well as others? So they brought their samples to the beamline and we popped them in. And we were able to show pretty quickly that the commercial samples were all almost 100% magnetite. So the highly magnetic form of iron oxide, which was to be expected. So that was good. We're on the right track. The UniMelb samples had a kind of a range of distributions, though, which is really interesting and useful to know. So it turns out the UniMelb samples were a combination of magamite and magnetite. Magamite being unstable and degrading over time. So this was consistent with what they had observed in the lab. So the UniMelb samples had a pretty wide range of compositions. The worst ones were about 80% magamite, but some were up to 100% magnetite. And then the others fell along the spectrum. So the group went back and analyzed the reaction conditions that had been used to produce these different nanoparticles and then correlated that with the compositions. And what they were able to show pretty clearly was that there was one salt that as they added more of that salt, they increased the amount of magnetite in the nanoparticles. So it decreased the magamite, increased the magnetite, until at some point, some concentration of this salt, it reached 100% magnetite, which is optimal for these nanoparticle formulations. So this was really fantastic results, just very clear, and it allowed UniMelb to go back to their industry partners and say, this is the recipe that works, and these are this is one of the really important factors. So that was really exciting. We were really happy to be a part of that. So X-ray absorption spectroscopy is only one of many experiments done around the synchrotron. So here's a kind of floor plan of our synchrotron in Clayton that I showed before. So this ring in the middle here is the storage ring. That's where we accelerate our electrons around. And then all these gray lines coming off of the synchrotron. These are all experimental hutches where we funnel the beam down into these areas to do different kinds of experiments. So there's 10 different operational beam lines, and they're really using X-rays in a hugely diverse and interesting set of ways. So there's tons of different research going on in here. I'll give you a couple of fun examples really quick. So we do have an imaging and medical beam line where they would, similar to a doctor's office, take X-rays, except the X-rays, it's extremely precise, extremely strong, and they can do 3D and 2D imaging. So one example of work that they did here, so they actually had researchers from Africa who were trying to make prosthetic horns for rhinos who had been victims of poachers. But rhinos are their skulls are very complicated. And so to get this right, to make a suitable prosthetic, they needed a really clear idea of what was going on in there. So they brought their rhino skull to the IMBL beamline and got this nice 3D picture, which helped them design prosthetic horns for rhinos. So it's pretty cool. Another fun example. So The X-ray fluorescence microscopy beamline uses X-rays to create elemental maps of objects. So it scans across an object and measures the elemental concentrations at each point. And here they've measured a Portuguese millipede. So you've probably seen these guys around your house. They're extremely popular or widespread in Melbourne. The synchrotron is just full of them. But so you can see that we've got 3 different elements here in different colors. So the blue is iron, the red is strontium, and the green is zinc. So you can see these nice little green blobs around the edge of the millipede. And those are her stink sacs, which is pretty cool. And then another really important beamline, I mean, they're all really important, but MAC was the crystallography beamline, which has been doing a lot of work on COVID-related projects this year. So at the crystallography beamline, they can solve the crystal structures of many things, but often biological proteins. So this is actually a protein from COVID, which they solved earlier this year, which could be used for therapy or just to understand how the virus works. So those are just 3 of the beamlines, but each one of these beamlines is doing just really exciting things and very different. Yeah, so we're really lucky to have the synchrotron in Australia. It's one of 70 in the world. It's a fantastic facility. I encourage anyone who's interested to look it up. And hopefully, once this COVID business is settled, we can welcome you back in for a tour. Great. So I would like to just first of all acknowledge that this was, while I'm presenting this work, this was not at all my research alone. This is very much a team effort. So this is Jess and Burnt are the other members of the XAS team who did a lot of this work with me. And then I'd like to thank the Mulvaney group and Nick for letting us be involved. It was wonderful to work with you guys. And I'd like to thank these 5 members of the Mulvaney group specifically, who did come down to the synchrotron and perform the experiments with us. It was fantastic having them. Yeah, okay, well, that's it for me. And I'll hand it back over to Josh, I suppose. Happy to take any questions.
Cool. Wow, that was fascinating. So interesting. I'll just get up the other slideshow. And that is right. We do have some time for questions. So hopefully you can see there's a QA button on your taskbar, I guess. If you could throw some questions in there, I will dish them out. We did get one earlier on, and it was from someone asking why silica shell specifically.
I'm happy to take that one. I guess that's relevant to what we've done. So the silica shell, I kind of briefly mentioned it. The DNA and RNA will selectively stick onto silica under the right conditions. So actually, if you add a special salt into the test kit soup or whatever you want to call it, the salt is called a chaotropic salt, which is a pretty fancy biology name, but let's just call it a special salt. That actually makes the DNA and RNA unstable, and they go looking for something to stick to, and they love to stick to silica. So they'll stick to that silica shell on those magnetic nanoparticles. I think this highlights one of the really interesting things you can do with nanoparticles, which is that they have a massive surface area, so that you dissolve them in a liquid, say, and then there's just millions and billions of them, and then you can stick anything you want on them, really, as long as you put the right surface coating on your nanoparticles. So you could use these magnetic nanoparticles to do a lot more than just separate DNA and RNA from something. You could, say you had a water source that was contaminated with some pathogen or a heavy metal or something toxic. You could probably design a magnetic nanoparticle with a surface that would stick to that nasty thing you want to separate out, and then just chuck a big magnet in there to suck it all into one spot and throw it out. So yeah, that's a really nice question, because I think it highlights one of the really versatile things you can do with nanoparticles.
Great. There's a few more that have come in now. got another question about the nanoparticles. Can they be reused for multiple COVID tests, or can you recycle them somehow?
Yeah, I guess in principle you could. The DNA and the RNA comes off the surface at the end, and then you sort of, they are back to the state they were in. Because of COVID test, though, you really need to be sure that it's not false when you get a positive or a negative test result. You want that to be as accurate as possible, because any inaccuracy there, you know, could have big implications, especially if you fact detect COVID when it was there. And so if you think about it, if there was just even the slightest chance that one little bit of like COVID was hanging on to the recycled magnetic beads, and then it goes into another test, and then it comes off in that second test, then that person will get a result that was meant for the original person. So that's why we wouldn't reuse them. They have to use fresh ones for every test, and that's why the demand is so high for them at the moment. But I'm sure for lots of other applications that didn't have such a large important outcome weighing on it, you could probably reuse them quite easily. Yeah.
Oh, very interesting. I've got a question for Valerie. You highlighted all of those different beamlines and all those different little kind of, I guess, lab spaces. How many, can you have like multiple going at the same time, or is it just one at a time, or like how does that process work? Do people have to wait? Is there like a big queue?
No, that's a great question. No, yeah, we run all the beamlines pretty much 24 hours a day, and then we usually take one day off a week to do machine studies or to fix things. Yeah, so yeah, we can use them all. I mean, it's just the electrons, or so the particles going around are just constantly spraying out a beam of light in all, yeah, so always accessible. Plenty of light to go around, yeah.
I'm just going to add to that. I mean, it's actually incredible, the synchrotron, like what the beamline scientists like Valerie are working, you know, shifts basically around the clock in some cases, because the experiments are always running. You know, middle of the night, there'll be scientists there running experiments. That thing never shuts down, or if it does, it's a big deal. So it's a pretty amazing facility.
Yeah.
And like you showed that rhino imaging of the rhino skull. How long does something like that take to actually be completed?
Oh, geez, that's a good question. I'm not sure about that one specifically. Yeah, I could guess, you know, a day or so, but I wouldn't know for sure.
And something like the millipede, which is more 2D and, you know, more comparative to a normal X-ray kind of, would that be a shorter time?
Yeah, I think that one was 2 hours, so that one wasn't too bad.
Cool. I've got another question here, and also a comment that you know, both Nick and Valerie, wonderful and very accessible talks. And I would like to second that. And then a question for Nick: could you comment on the hurdles that you faced when going from 50 mils to 50 litre batches, and can you continue to go up from 50 litres?
Yeah, that's a thank you for starters for the kind words. It's a great question. This was pretty uncharted territory for us to scale up a reaction so much and so quickly, because we were really racing to try and prove that it could be done at scale. Otherwise, there wasn't a lot of point. Big problems along the way. I mean, one of the things that happens when you just take something and try and make it huge is that, I mean, to go back to pasta sauce analogy, you can imagine if you're putting heat on the bottom of your pasta pot, when it's really small, you can just kind of stir and nothing will stick, hopefully. But if you were using a really big pot, like, stuff is just going to stick on the bottom, and there's no way you can get a big enough spoon to stir that thing quick enough. So just things like this come in where you realize that the way that you heat liquids and move things around, even just pouring chemicals into a reactor. You know, we had, I think I showed a photo at the end of my talk with one of our students had to be wearing like a full respirator, because, you know, if the fumes coming out when you're dealing with 1 milliliter in our lab is not a big deal, but when you're dealing with 2 or 3 liters, it can be a problem. And then in terms of what happens, I mean, you scale up, you think you've got it right, and then you go and look at it on a microscope or in a COVID test, and it doesn't perform the same way because something has changed, some variable has been affected by that scale up. You know, for example, like, you know, in Valerie's talk, she was mentioning how we had this salt that was affecting whether or not the composition would go one way or another. If we went the concentration we originally had this salt at was kind of the tipping point between the two, and as we scale up, it'll fall one way or the other just based on kind of random chance almost. So lots and lots of problems like this, but I mean, as always with science, you just kind of have to keep testing and keep at it and just chip away until you solve all the problems and hopefully have something working at the end.
Just a lot of trial and error. Are the nanoparticles that have been produced, have they been used in tests already? Or is it more a trial to see if it could be done?
It's still in trial. We're not making, it's not in production, I guess I'll put it this way. But that would be the next step. So essentially we have what you might call something like a minimum viable product or a viable product where you've shown that it works, you've demonstrated that at an appropriate scale you can make, you know, enough of these things to be relevant for the problem, in this case COVID testing. And now we're working with the industry to try and get those agreements in place that someone will make, you know, this much of it and someone will buy this much of it. You know, these problems are less scientific in nature and still quite interesting.
And this is a question, I think, coming off the recycling of nanoparticles question before. Is there an environmental impact of the production or the, I guess, disposing of them as well afterwards? What does that look like?
This is a great question. Yeah, obviously anything with the word nano or nanoparticle or whatever in it, you know, we have to be careful with safety. And as researchers in the field of nanotechnology or nanoparticles, it's always the front of our minds. So I guess I would say that I think I kind of already used the analogy before that you could use magnetic nanoparticles. Are quite easy to move around. You can just use a magnet to collect them. So I wouldn't be too worried about losing them into the environment. But in general, we're going to focus on non-toxic materials for applications like this. So iron oxide is not toxic. I mean, the desert in Australia is made up of iron oxide. And the other thing I'd say is that a lot of these applications we kind of mentioned that the Exciton science works on, things like, you know, solar windows and stuff like this. When you're putting nanoparticles into a piece of glass or into a piece of plastic, they're really stuck in there. I mean, we couldn't get them out even if we wanted to once we've embedded them into our window or our LED or solar cell or whatever. And so there's really not a risk once you've put them in the application that they're going to escape or anything like this.
Cool. That's good to know. And I guess sticking with that topic a little bit, obviously, this was there was some of the stuff was used in the development of solar technologies. Going back the other way, we have a question here. How would you expect that the expertise from this example of industry engagement will impact the projects on solar energy?
Yeah, good. That's a really good question. One of the nice outcomes from this project from us, beyond everything I've discussed today, is that where we learned so much about scaling up chemical reactions and working with industry. We actually had a solar energy related industry project, which I mentioned to do with solar windows, pop up. After this, we'd been working on this magnetic silica project for a while, and because we had the experience sort of under our belt, so to speak, in scaling up reactions and working with industry, it was much more comfortable for us as researchers to then say, yeah, we can do this, and we can scale that reaction up. Where we know how to do that now. It's something that we're much more confident that we understand the problems and whatnot. So it has directly fed into our attempts to work with industry relating to solar energy. It's a really good question. Thank you.
Sweet. Well, it has just hit 7 o'clock now, so I think we will wrap things up. Was there anything, any last minute comments that either of you wanted to add before I just do some thank yous? Cool. I'll take that as all good. To be honest, I did not expect such a breadth of analogies, and really great ones, from cooking pasta to corgis. Wow. So I think that comment about very accessible talks was spot on. So I did want to say a huge thank you to both Valerie and Nick for jumping in here into this public lecture, which was a bit of a COVID digital pivot, but such a great topic, and yeah, obviously something that is very relevant to our lives right now and very important. So thank you also, audience, for coming along and listening in, and also asking some really great questions too. If you want to find out more about Exciton Science, what we do, there's our website, there's our handle, and I'm sure there's also a bunch of interesting stuff on the ANSTO website too. So yeah, thanks for wrapping up National Science Week with all of us, and I hope you all have a fantastic evening. Thanks, everybody.
Thank you, everyone, for joining.
Yes, thanks. Have a good night.